for any living organism. There are some variations there at the bottom. They deal with pressure and salt instead. But we've got cardinal temperatures up here at the very top. And these are cardinal conditions. Almost everything has an optimum condition that they will survive at the most themselves. They will be able to produce the most progeny. But they also have a maximum survivable limit. And then they have a minimum survivable limit. And we'll get into these more later. It's not something that we're just talking about today and then gone. But each organism has their own specific cardinal temperature or cardinal pH or cardinal pressure. A scope of three conditions that they can survive at that minimum, that optimum, that maximum. This is something we talked about in lab already a little bit. Remember, some organisms like bacilli, they love 45 degrees Celsius. That's where they thrive. Where something like Acaricines with the red pigment thrives at 28 degrees Celsius. So these are all things used to put organisms onto the tree of life. This is a figure from your book, so if this is small or grainy in this presentation, you can definitely go to your book and look at this. But this is just a few of the examples of different phylums of bacteria. There's actually 23 phyla of bacteria, so we don't have all of them on the slide. But the ones that are on here are typically the most important, the ones that we interact with the most. And they're represented by the size of the block, by how many individuals are in that group. So hopefully some of this looks a little bit familiar from your paper with the oral bacteria. Remember, the oral bacteria tended to be more gram-positive. That's that little tan block up here, where the intestinal bacteria niche tended to be more proteobacters. Well, your proteobacters are another name for gram-negative things, or thin-skinned. That's the largest group we know about. Now, it is a very real and likely probability that we know about more of these because more of them make us sick. It may not be that there are really more of them out there than, say, the gram-positives. It may just be, and it's very likely, that we just know more now and have cataloged more now because they're ones that have more significant illness associated with. Now, there are so many proteobacter or gram-negative things that have actually been subdivided down into alpha, beta, gamma, delta, and epsilon groups. And they talked about this in the nanobacteria case study as well. So this is the third time we've seen this pop up. So be familiar with that terminology because we're seeing it over and over again. Your case study compared nanobacteria to a proteobacter group, or suppose that they might be into the proteobacter group, which is a gram-negative that causes lots of disease. We also have the firmicutes. This is another name for gram-positive. You'll see that in different literature. And then we have the tenricutes, or the mollicutes. These are things that lack a cell wall. A lot of these mollicutes actually cause urinary tract infections and things like that. You'll see this very quickly in an upcoming quiz when it talks about mycoplasms. Not to be confused with the mycobacterium. Mycobacterium are your tuberculosis, leprosy kinds of diseases, whereas mycoplasma tend to cause UTIs. They also, Mycoplasma pneumoniae cause pneumonia, obviously, hence the species name there. But those things don't have cell walls, and so you can't attack them with an antibiotic that targets the cell wall. So those are some things to think about. That also ties back into the terminology at the top of the slide, the Berg systematic bacteriology. So there's two different Bergies. Bergies is the guy who put together a book that helped to discuss and break down the phylogeny that we see with the bacteria. The systematic is what we use for diagnosing somebody. If we cultured some kind of cut, somebody had a cut on their skin, we cultured that on a mannitol salt plate, and we're looking for the media to turn yellow or change. That would be, and then if the media changed, we said, oh, that's a Staphylococcus aureus. That would be a systematic approach. I applied it onto some media, and from that media, I make an assumption about what that bacteria is. Berge systematic is the most common bacteriology test that you'll see in a clinical setting of microbiology because it is all based on systems and reactions to biochemical tests. It's not really concerned with necessarily phylogeny or the evolutionary relatedness. That's where the Berge determinative bacteriology comes into play. The determinative is almost always genetic based. It's looking at how many base pairs differ between these things. It's great as a benchmark for really figuring out where these things go down to the minute level, but from a clinical standpoint, it doesn't matter as much to clinicians, which is fine. If I'm a clinician, I'm not going to do a PCR test to figure out if the kid's got Mycoplasma pneumoniae. I'm going to grow it on a plate. I'm not going to do a PCR test. It's going to take too long. It's going to take too much money to figure out if the kid's got Staphylococcus aureus. I'm going to grow it on a mannitol salt. So systematic is what we'll be more interested in throughout the course of the semester. Here's a couple of examples of the domains of archaea. Now, archaea are a different thing. There's 3 major phyla of those, and then those phyla are broken down to these families and orders, classes we see here. So these are 2, the Euryarchaeota and the Crinarchaeota are 2 of the 3 main phylums. We're going to talk about the third because when we have less interaction with it, then we do these Euryarchaeotas and Crinarchaeotas. But things to think about with the domain archaea, they don't make us sick. There is not a systematic approach separating archaea because they make us sick. All of the archaea determinations are of the sequence variety, of the determinant variety. And DNA is the main thing we use, or ribosomal RNA is the main thing we use for classifying these. The 2 major phylums that you kind of think about are the Euryarchaeota. We know more about these. It's a more well-developed. It's the methanogens, the halophiles, the thermophiles, the things we saw in that previous slide, and really, really, really look a lot like the mollicutes. And if we go back to that first slide with the phylogeny, we'll see that the hyperthermophiles and the mollicutes are actually closer to the tree root, which is closer to the archaea, than something like the proteobacters, which have the gram-negative, the cell wall and the outer membrane. The Crinarchaeota tend to be oceanic. There's probably a lot of them out there, but the oceans are so vast and we can't access them very well that we just don't know what all is out there. Also, your hyperthermophiles are your Crinarchaeotas. A lot of your soil and freshwater archaea are also of the Crinarchaeota persuasion. Again, man, the main thing to know about archaea is they don't make us sick. We don't tend to study them as often. Now, with them being closer to the root of the tree life than the eukaryotes, we do care about them in that regard, trying to understand our own origins. And archaea actually literally is derived from archaic or old. These are old cells. If you think about, methane-rich areas,
salt-rich areas, high temperatures, low temperatures, extremely high temperatures, those conditions mimicked early Earth 3.5 billion years ago. These probably are some of the older organisms, and it's not that they've changed to survive in these conditions. It's probably they developed in these conditions. As those conditions became more moderate, they found niches that resembled old Earth with the high methane and high salt and high temperatures or low temperatures. And they are really kind of a neat into our own past, but beyond that, they don't make us sick, so we won't spend a lot of time with them. And then the last thing to talk about for this chapter are the eukaryotes. There are only 2 groups, the protists and the fungi, that will have a lot of interaction with through the course of the semester. We'll briefly discuss helminths and lab, the small worms, which are animals. But for the most part, the protists and the fungi are the things that are really small that we'll have interactions with. Remember, the main thing that puts them in this group is they have a nucleus. After that, it really doesn't matter. They may or may not have any one of the other organelles we've talked about. not be able to do some of the more complex things eukaryotes can do. They really need to have is a nucleus. That's what puts them in that club. So a couple of review questions to think about. What is purpose of staining in light microscopy? What we're really looking for is differentiation and contrast. These things are so small, so very, very small, but we need to have some contrast so we can tell and make assumptions about their structures. Think about what would happen if you took all the plasmid DNA from the prokaryotic cell. What I see is that there would be side effects. I could take all the plasmid DNA out and it wouldn't do anything. Think about why that's different from removing the housekeeping chromosomal DNA. I'll let you guys think about that question on your own. And then also another big question kind of thing thinking about is there are probably a lot more microbiotic organisms that we have not discovered than we have discovered. And think about some of the whys there. Think about the archaea especially, when we were just talking about that. Why don't we look at some groups? What are some of the roadblocks to looking at groups like archaea, especially if their interactions with us aren't especially noteworthy? So those are some of the questions to look on, some things to be thinking about. Your review is already online. You'll have a test on Tuesday. So be sure you listen to these lectures. Hopefully you've already started studying because there's a lot of material, even though it seems like just a few chapters. There will be 20 multiple choice questions on the test. You'll also have 5 matching. Those are going to be stored figures. So look back at the stored figures from chapter 1. You have that there. You'll have 10 fill-in-the-blank questions. Those are terminologies. Look at the terminologies discussed in class predominantly. If I didn't talk about the terminology, I'm probably not going to be hung up on it. But there was some terminology that we've outlined, defined, discussed through our lecture, so be sure you focus on those. You'll have 5 short answers. Short answers are usually true, false, yes, no, perform a calculation. Something that's pretty straightforward answer, but then you have to explain your answer. Give me a 1-sentence rundown. Why you think this is false, or why you think this is true, or why you did that calculation. You'll have 4 of those, and then you'll have 3 essay questions, 5-point essay questions. Just kind of look over the material, look at the review, listen to these lectures, and if you have any questions, I will be in the office Monday, and Tuesday morning until 9:30. So, happy studying.